Welcome to the Peer2Peer Alumni Showcase Webinar, everyone. We will be getting started in just one moment while we wait for people to trickle in. All right, looks like our attendees are going up, so I guess we will get started. So welcome everyone. Thank you so much for joining us for today's peer-to-peer webinar. Please note that this session is being recorded, so you'll be able to come back and see it at a later date as well. My name is Carly LaRoche, and I'm the coordinator for the peer-to-peer program and a member of the GOA-ON Secretariat. This webinar is presented by GOA-ON, the Global Ocean Acidification Observing Network, and NOAA's Ocean Acidification Program. If you would like to stay up to date with the GOA-ON community, including the peer-to-peer program, you can consider signing up as a GOA-ON member. We'll be sharing the membership link in the chat for those who are interested. And today we're really excited to be joined by two peer-to-peer scholarship recipients who will be sharing about their research experiences with the peer-to-peer program. Peer-to-peer is an international mentorship program that pairs mentees who are new to ocean acidification work with experienced mentors in the field. The program aims to foster community among ocean acidification professionals, build long-term capacity to measure and address ocean acidification globally, and provide training opportunities to support careers in OA-related fields. Since it began in 2016, Peer2Peer has supported over 250 participants from over 60 countries. If you are interested in participating in Peer2Peer, either as a mentor or a mentee, we will be accepting applications to join the next cohort starting in July 2026, and we encourage you to check out our website to learn more about those upcoming opportunities. Another piece of the peer-to-peer program is we partner with the Ocean Foundation to offer scholarships that facilitate collaborative projects between these mentor and mentee pairs. For any active peer-to-peer participants who are here watching, there is an open scholarship opportunity now with applications due March 31st. So you can reach out to me or see the peer-to-peer website for more information on that. And a couple of words on housekeeping before we get started with today's webinar. During the presentations, all participants are going to be in listen-only mode, and you're welcome to type any questions into the questions box, which can be found at the bottom of the control panel on the right-hand side of your screen. We'll be monitoring these incoming questions, and we'll pose them to our speakers during the question and answer section, which will happen right after each of our speakers. For the discussion, you can also use your raise hand function in the toolbox at the bottom of your screen, and we can call on you to ask your question directly. And with that, I'd like to introduce our first speaker today. Dr. M. Anand will be presenting ARMS, Autonomous Reef Monitoring Structures, an emerging tool to monitor ocean acidification and coral reef health. Dr. M. Anand is an associate professor and the head of the Department of Marine and Coastal Studies at Madurai Kamaraj University in India. His pioneering research on ocean acidification, coral reef resilience, and marine biotechnology has earned him international recognition and collaborations across Asia Pacific and beyond. Dr. M. Anand is a peer-to-peer alumnus 2023 to 2024 and collaborated as a mentee under the mentorship of Professor Dr. Aileen Tan, Director of the Center for Marine and Coastal Studies at University Sans Malaysia. Thank you for joining us today, Dr. Anand, and I will hand it off to you.
Thank you, Dr.
Nice.
Carl, for our nice introduction and coordinating this webinar. Can I share my slides, Carl?
Yes, you could share your slides.
Is my screen is visible?
Yes.
Okay. So, very good evening, good afternoon, and good morning to one and all in the different time zones of the participants. I'm Dr. M. Anand. I'm happy to uh, share my experience as a peer-to-peer alumni of uh, GOA-ON. And I thank GOA-ON and the Ocean Foundation and my mentor, Dr. Aileen Tan Shau Hwai, for giving me this opportunity to take up as a alumni of this peer-to-peer. During this program, I acquired some information on about the arms. Arms is not new to our ocean acidification research, but arms use with reference to the aragonite information and the biological response is what we found it's new to the acidification research. And this technique we got learned from the lab of our mentor, Dr. Aileen in the CMAC. And I'm going to present my experience, what I acquired some knowledge, and what may be the future research in these aspects of it. The slides are not moving, right?
Yeah, we're not seeing them move.
Wait. I'll do one thing. I'll switch off my video so that buffering can be better. Yeah, is it moving now?
Yes, that's going now.
Yeah. So the journey started with my letter to Dr. Aileen requesting permission to work in her lab. And the the biggest challenge in uh, winning this competitive scholarship is you have to identify the right mentor. Because once you identify the right mentor only, you can write a very good proposal and you can identify a the right research question. So in that aspects, I wrote to Aileen to learn how to fabricate the arms and how to deploy the arms and how to analyze the results of the arms and all these things within mind. I wrote to Dr. Aileen, and one of the news article which I could come across when I was searching for the mentor is that Aileen has done the first ocean acidification monitoring station. She has uh, started this station in the South China. And this news has provoked me to learn more about these arms, how these arms could be a monitoring tool for ocean acidification research. So that part of the research problem has made me to write to Dr. Aileen, and she readily accepted. Uh, when when I written about what type of techniques I wanted to learn from her lab, I elaborated on the what is the background of us and what research gap we wanted to learn from her lab. She readily accepted, and we got the consent from her. So I should thank Dr. Aileen for giving this opportunity. And what I returned to Aileen is, what is the skill set we already have? That is for the since 2010, we have been doing some microcosm experiments on various calcifying organisms. And recently in the sense, by 2021 and 22 onwards, we started monitoring the aragonite saturation state in the Gulf of Mannar and Park Bay regions in the South India. So two different aspects. One area we are very strong in perturbation experiments, and another area we started how the aragonite saturation state can be a signature for ocean acidification monitoring. So between that, what we lack is how the biological organisms responds to it. So that is what we wanted to identify in as a research gap in this particular proposal or the project call. And we could luckily win this call, and I got paired with Dr. Aileen as a mentee. So what about arms? Arms are, as I told you, it's not new to acidification research. We all know that it's a stackable plates of artificial structures that mimic the natural reef community. So once you deploy these arms near the coral reef region, the organisms will start colonize, will got settle in these arms, so that we will get the information about the organisms distribution, the diversity, and if you could be able to collect these information at different sites and different period of time, so we'll get more details about how these cryptic organisms are responding to the changing chemical signatures as the acidification stressors. Is the slide is moving?
Not moving forward right now, but maybe we can give it a second to move.
Yeah.
Yes.
Now, can you see the bridge the knowledge gap? Yeah.
Yeah, we can see that.
Yeah. Yeah. So, what particular research question that we wanted to identify in this particular study was, as I told you, we already done a lot of microcosm experiments in terms of perturbating CO2 in the controlled experiments. And similarly, we already have data on aragonite saturation level. So the biological organisms response, how they react over a period of time, and at what is the succession level when there is changes in the aragonite parameters. This is what we wanted to identify. So for that, we wanted a field-based monitoring for which the arms play an important role. This is where we bridge our knowledge gap. And once this arms has been deployed, we could be able to identify the acidification parameters of aragonite saturation state, alkalinity level, the pH values, salinity, temperature. All these factors will give us an ultimately the value of aragonite saturation state. And the arms factors, from the arms factor, what we could understand is we can linkage how the predictions, what we did in the perturbation experiment, is that match with the natural condition. So in terms of biomarkers, in terms of DNA methylation, in terms of calcification factors. So all these things we could be able to understand. So this is what we have done and in our study. This is about the arms materials. Arms material is very simple. We have a PVC base plates, And these are nothing but the plates are arranged in a regular space with many cross spacers and washers, bolts, nuts, everything. And it's very simple, user friendly. You have in this website the Global Arms Program Protocol. One can go through easily, and you can make it a local fabrication also. Similarly, we have ordered from a local agent the PVC plates with all the crossbars, necessary uh, accessories for this arms. We brought the plates and we assembled the plates in our lab before going for the deployment. This is the place where we deployed the arms. This place is called Pudumadam and Valangamburi. Two places we have deployed these arms. Actually, the study site is a Gulf of Panna region, which is a marine biosphere reserve region and is known to harbor 3,000 to 600 species of flora and fauna. We have rich coral reefs distributed in this region, and adjacent to this is the Park Bay. This region also contributes a lot of diversity and supports the diversity of Gulf of Mannar region. And this is the place where we deployed the arms. Once the arms is deployed, we have to give a sufficient time. It depends on the research question what we are going to address. Since we wanted to gain the knowledge, we wanted to gain the experience training of it, and also we had a very short period for this completing the study. We deployed the arms and we have given the time of 46 days to retrieve it back. So once we retrieved, we have photographed the naming and labeling is very, very important. And as we extract the organisms, we also taken care to store it for further sequencing, genome sequencing. Basically, the main structure, the main result, what we obtained with this 46 days is we have observed the arms plate in the two different intervals. That is one with the 20th day and then another time is 46th day. So you can find from the results from when we observe it in the 20th day, it was mostly dominated by bare plate. And you can see the ecological succession very clearly in this, for starting from biofilms is dominating, then cyanobacteria, fungi, then bryozoans, cytozoans, and the started slowly colonizing in the 20th day. At the end, 46th day, when we retrieved the arms, we could find the biofilms was there, but equally you can find the succession that cryptic organisms, the grazing organisms are started dominating the plates, and followed by the shrimp larvae, which was seeking the shelter in the arms, and finally the seaweed patches. Seaweed patches, here I wanted to mention, the place where we deployed is called a seaweed paradise, the Pudubadam village. So, I mean, at the 46th day, we could find seaweeds, not much of coverage, but we presume that if we leave this arms for another 1 year or 2 year or something like that, we could be able to see the succession very clearly. Ultimately the seaweeds dominate the entire plates. As we took the samples, we have done sequencing. One point I wanted to make a note here is uh, there is the scope for this study is enormous, but within the given duration and within the budget, we wanted to work on it. So we could not be able to go for sequencing of all the organisms what we took from the samples. So selectively we took three amplicon libraries with molecular markers of koi gene, and we could able to find Cleona, Diadinum, and Aplysia as three organisms we could able to get from the sequencing upon the blast result. This shows that the organism, I mean, the place is very diverse. We had lot of diverse sequences, but we have preserved it. We wanted to do that sequencing in future and find the diversity. One point here is we have done this observation. Once we have deployed the arms, we started collecting the aragonite parameters level, as well as at the end, during the 20th day and 46th day, we started observing the settlement of the organisms over the plate. What we found is the chemical signatures and the biological response was highly correlating. When we had the alkalinity level as well as the aragonite saturation level was varying, we could find a lot of changes in the plates deposition of these calcifying organisms. Since we have few data of the sequencing and the aragonite values, I could not able to give you the entire details of it, since we are going for a publication part of it, and doing little part of it is left to go for analysis. So that correlation was existing in this study. When there is changes in the aragonite saturation state, the cryptic fauna settlement is also varied with both the regions, that is the Pudumadam as well as the Velangapuri. The ultimate aim of the study is for the capacity building. So we wanted to actually the arms usage in India is very, very limited. A agency called NCSM, which comes under Ministry of Environment and Forest, they started deploying it, and they go for varying modifications of it to studying it. In their website, they have given some details about the usage of arms. Other than this, there is no publications which could be able to support with some data. So what we wanted to present this in the national seminar. Similarly, I've done it in Ocean Society of India conference, which is held in Goa. I could be able to get a lot of response from the scientists, and a lot of informations we shared, and this has given some sensitizing information to the persons who are working in acidification research. And similarly, once our mentor has hosted a program on POGO annual meeting, we showcased our findings in the POGO 26 annual meeting with a general picture about the Gulf of Mannar and Park Bay region, the status of acidification research. In that, we have given our previous studies results on perturbation experiments, aragonite level. We can see in this graph the aragonite levels are given in a map, how the Park Bay and Gulf of Mannar is varying. And the key results, what we achieved out of the study, the scholarship, is we ourselves has got trained with assembling and installing and retrieving and photographing and studying the results of the arms. And apart from this, we have started a regular monitoring in two sites as a PhD thesis work. One of my students started working on it. And a national level workshop, we have trained with 75 participants. The arms results were presented in OSI conference and POGO 26 meeting. The stakeholders meeting was also organized, in which the mentor, Dr. Aileen, has joined with us, and she explained very clearly to the scholars, all the stakeholders who attended the meeting. And the DNA extraction and sequencing has been done, and slowly we are doing for few species as we meet the budget. And another part of this peer-to-peer program is the capacity building, one part, and another is the dissemination of information to the local people. So a lot of local news channel coverage has been done, and also a lot of information has been given in the local newspaper to sensitize this information about the usage of arms, because the use of arms is not only for research. The arms will also, if we could able to deploy the arms in a large scale near shore, we could able to attract lot of juveniles, brooders to take these places as a shelter and a feeding ground. So that part has been sensitized to the fishermen, and it was more. I mean, the fishermen were more welcome about this move, and we wanted to take up this further to the local bodies like fishermen organizations, the ministry, to take up this research further. In a long-term study, as I told you, one of my research scholar, actually, as I told you, the study area is a protected area, biosphere reserve region. So, formally, we submitted a request to the Ministry of Forest, and they given after a presentation, they given us some recommendations, and with the recommendations, they sanctioned us to allow for only 2 units of arms to be deployed. But again, we wanted to represent to the community to at least 1 unit be increased in an individual site, because for the statistical reason, we want at least 3 arms to be deployed in the same site. So that Mr. Johnson of my lab, he started working on this to identify how this biomarker response and acidification changes can be done in the bivalves. So what are the challenges and solutions in handling arms is the monsoon season. You have to have a clear idea about when the sea is going to be rough and when it will be calm. So in our area, we had a difficulty in southwest monsoon, so that it made us to delay of deploying our arms. That is the one part. And the budget limitations for DNA sequencing. Lot of scopes is there in this as you get a lot of samples, but DNA sequencing requires a lot of budget. So that we have to keep it in mind. And another part is administrative delays, as normally it happens because the money transfer happens, it has to be university approval has to be done. Due to this, we had a 2 months delay, and luckily I could able to get the 2 months extension from the GOA-ON, so that I could complete the results as what is expected for the study. And before I conclude, this is about our department. Our department is in Pudumadam, a coastal village near to the Gulf of Mannar and Park Bay region. But our main campus is in Madurai. We call it a temple city. From Madurai, it is around 140 kilometers to reach our lab. Our lab is a marine field research facility. Actually, it is since we have a very limited facility, we could able to do the wet lab experiments over there. We collect all the samples, we do all the basic processing over there, and we bring it to the main campus where we have a lot sophisticated instrumentations for analysis. Basically, we have analytical lab and a good facility for seawater pumping, and scuba diving facilities also there. As I mentioned earlier, the Pudumattam village is called a paradise of seaweeds. So we also do the seaweed farming in our place, and we already done that entrepreneurial training. And apart from this, we normally do the International Coastal Cleanup Day every year since 2013. We have taken up in an annual movement where students, college students, school students, fishermen community, all they participate, and they do cleanup activity in the Pudumadam village. And this is for the yearly career researchers and students, those who wanted to join as a mentee for this peer-to-peer program. You are very most welcome to our lab. We have a basic facility for microcosm experiments. We have done with organisms of calcifiers. Near our lab, we have bivalves, echinoderms, a lot of diverse organisms are there, so that you can use these organisms to study how these organisms could be respond to acidic conditions. And also, not only the lab-based research, if you wanted to do some ecosystem-based study, you can almost welcome. Where we have a lot of resources in our labs, near our labs. It's a very rich mangrove ecosystem is there, and coral reefs are there, seagrass, seaweeds. So a lot of resource, or you can see all the ecosystems which are more critical ecosystems of the marine environment is near the vicinity of our lab. And coming to the end, even the mitigation aspects of this acidification also can be studied if you visit our lab, because around our lab we have a lot of seagrass ecosystem and mangroves ecosystem. Recently, we published a paper in Springer Nature, in which we have highlighted how this spatial dynamics of aragonite saturation state and the blue carbon stock meadows of the Park Bay is could able to be a mitigating solution for acidification in near Gulf of Mannar and Park Bay region. So at the acknowledgement, I thank my mentor. I think my mentor is, I'm requested her to be online. If she is online, I thank you, ma'am. Thank you for being my mentor. And I thank GOA-ON and the Ocean Foundation for funding this study. So thank you so much, Carol.
Thank you so much for your presentation, Dr. Anand. That was wonderful. It was really great to hear more about your research, really fascinating results, and also great to hear that it kickstarted some long-term monitoring stations. I hope that some of the peer-to-peer mentees this
Yeah,
year or in the
mentees
future
can
are
be
able to
welcome.
work with you. Yeah.
Sure, sure. We welcome
Um,
mentees to join here.
yeah, wonderful. So at this time, we're going to open up the floor for questions. And so you can feel free to put those in the questions box at the bottom right hand of your screen. And we actually have a couple coming in already. So the first question is from Ilaria Dianelio. Thank you for the interesting presentation. I have a question about the arms deployment time. Is there a specific reason for choosing 1 month rather than 6, for example? Is it dependent on the site you focus on?
No. Actually, this is not depend on the site or whatever it may be. But to be very frank, the time constraint only we have deployed the arms, and we could able to retrieve within the 46 days. So if the time permits, because we know that peer-to-peer program, it's given for 2 years, right? And with all the administrative formalities and all, and once we start the work, it will be almost 1 year is over. So that is the constraint. But since it was a training opportunity, we did it for 46 months. You can do it for multiple arms. You can deploy, you can retrieve it every 3 months, 6 months, or 6 months after maybe 12 months. or 24. Like that, you can partly retrieve the arms so that you will get a very good detail about the succession. And the
Excellent.
next question is?
And the next question we have is from Marco
Marco.
Munari from Italy. Great talk, thank you. I was wondering if you considered the effect of light on arms colonization, specifically whether you observed differences between the upper panel and the internal plates, and if photophilic versus sciophilic taxa responded differently across the sites with contrasting environmental characteristics.
Yes, it's really an interesting question, Dr. Marco. Actually, this is right. Actually, uh, light aspects is very much important because we have been deploying in a very shallow region, these arms. And as I told you, our place is rich with seaweeds. So we could find in the upper panel a lot of seaweeds are got deposited. And in between that, we could find the tunicates and a lot of ascidians or things like that, which uh, seek the hiding behavior. We could find it within the gaps. So this is, of course, when you see the natural rocks, the same sort of succession we can find it here. The light part is very, very important. If suppose you deploying the arms in the much more depth, then you will not get the organisms which are being more influenced by the photoic response. So that way, the researcher has to decide what is the depth you wanted to deploy. You can aim for deploying at a different depth so that you can get a succession of these organisms in very clear.
Excellent. Our next question, well, first you have just thank you for the interesting presentation. Congrats on the achievement. The next question is from Alejandro. What is the required spacing between each plate? And when anchoring them, how deep should they be set?
Yeah, that is very important. Actually, the spacing is very standard in the arms protocol. It is there. We can easily find 1 to 2, 1 to 1.5 inches is there. And the another part is anchoring is very, very important. We could find the anchoring depends on the local sea condition. If the sea is much rough, you should have to make it nail it with the hard surface. And similarly, when you nail it, when retrieving back is very, very difficult. So depends on the local condition, we have to decide the anchoring facility. Ours, we had a bad experience of first anchoring with the stone. But since it's a rough condition, we lost one arms and we could able to do it again. So that is what we have. We will get some lessons out of it when you did not understand the local sea condition of it.
Yeah, that's very good advice. Our next question is from Ashanka. Can you explain a little bit more about laboratory mesocosms? What are the pH conditions that you are maintaining, and how you are monitoring changes of organisms there? Thank you.
So actually, that is not a mesocosm. It is actually a microcosm, what we are maintaining, right? Mesocosm is what we need to do it in the wild, I mean, in the controlled condition. A part chemical factors or physical factors you can control. But completely what we are doing is a microcosm experiment. Microcosm, we have a perturbation experiment. We have a CO2 cylinder. We purge carbon dioxide into different concentration of at different concentrations to maintain the pH of 8.2, 7.8 and 7.5. It depends on the researcher because previously we have done with echinoderms, we varied it with say 8.2, 7.8 and 7.5 and even 7.2 we have done. So depends on the researcher, 3 level of pH they maintain or 4 level of pH they maintain for the study.
Excellent. And I think we have a lot of questions in the chat, but I think we have time for one more. So I'm going to pick one from Amel Balaj. Did you do sequencing for the diversity monitoring or for another purpose? Is there any specific direct relationships between species and acidification?
Absolutely. This is a great question, Amal Belraj, I think, right? Amal. So actually, this question, this is what will give a a genetic signature of your acidification research. This has to be taken up worldwide. The calcifying organisms will respond in terms of DNA methylation. So if you could able to study the calcifying organisms, the methylation aspects of it by sequencing, you will get a signature of it so that when you wanted to do a vulnerability study in a particular ocean or something like that, you can collect some organisms, check with the DNA sequencing or signature of the, if you have a metadata or a database, we can easily compare of it. So it's a long way to go, but it is the next area of research we should focus on.
Excellent. Thank you for your responses to those, and thanks everyone for your really thoughtful questions. This was a great discussion. Um,
Yeah.
if I know that there are a couple of people who weren't able to have their questions responded to, so I'd encourage you to reach out to Dr. Anand and
Yes.
keep the conversation going. So with
Yeah.
that, thank you so much for joining us today, Dr. Anand. I know that you
Thank
need
you.
to get going,
Thank
but
you. Thank you. Thank you.
yeah.
Thank you, Carl. As I told you, I'm on the move. I'll switch off my video and I'll be in the line, right?
Yes, sounds great. Thank you.
Thank you. Thank you. Thank you. Bye bye.
All right. So at this time, we're going to move on to our next speaker. Um, so let me share my screen for my introduction. All right. So our next speaker is Dr. Georgina Cepeda, who will be presenting on planktonic calcifiers under pressure, implementation and results from ocean acidification training in the Southwest Atlantic Ocean. Dr. Georgina Cepeda is a researcher at the National Council for Scientific and Technical Research, working at INIDEP's zooplankton cabinet. She has a doctoral degree in biology with a strong background in biological oceanography. Throughout her career, she has specialized in the ecology of marine zooplankton, becoming a young leading researcher in diversity and ecology studies of zooplankton in the Southwest Atlantic Ocean because of her outstanding contributions in the discipline. She is part of several INIDEP projects, which include among their study variables the discrete estimation of the carbonate system variables. Dr. Cepeda was awarded the Peer2Peer Scholarship in 2025 in support of her collaborative work with her mentor, Dr. Nina Bednarsek. Thank you for joining us today, Georgina, and I will hand it off to you, and I'll share your slides as well. Okay, looks like they are up, and I think we're ready for you. Yeah, we're having trouble hearing you, but I know that you're having some internet challenges, so hopefully we can get it working. Just hold tight for a minute while we get this working. All right. Well, while we're waiting, I will give a little brief shout out to some of our partners that we've been working with peer-to-peer. Oh, hopefully. And which has been, we've worked with the Ocean Foundation to kind of facilitate these scholarships, and we're really appreciative of their support in helping get these through. We also have you see Carla on the screen there helping out. We have been working a little bit with people at OA Cookbook, which is the practical best practices for ocean acidification monitoring. And so this is going to be one of the future webinars, potentially for peer-to-peer, where we'll learn a little bit more about these practical best practices. And there's a resource, an online resource that you can use for that. And it looks like we're still having some audio issues, but working on it. Can't hear you still, Georgina. Oh, that might have worked. No, no, can't hear you. No. Let's see. All right. So I think if we're Georgina, could you try connecting just via phone? I think that there's in your panelist link that you got, there's likely a just like a phone number that you can call to connect to audio. And thanks to our participants for waiting. We'll try to get this sorted out in the next couple of minutes. Yes. Yes, we can hear you. Could you say something one more time? All right. Yes, I think that we can.
Okay. So, hello to everyone. Well, my name is Georgina Cepeda, and it's a pleasure for me to share with you the outcomes of my Pier2Peer fellowship from the last year. I want to start this talk by thanking the Ocean Foundation and the GOA-ON for making this experience possible. This project was a truly international effort since my mentor, Dr. Said, she's from Pesquero, and I am from Argentina, but we met in the US to do our activities. So we have an intermediate fancy to meet. The title of my presentation is Planktonic calcifier under pressure, implementation and results from the ocean acidification training in the title. Okay, the main goals of our research plan were twofold. First, to enhance our understanding of the impact of the ocean acidification in the calcifying of plankton in the ocean. And then, and the other one, to transfer knowledge and expertise related to the methodological approaches allowing for detection of surface shapes. Well, as a study subject, we focus on ecosystem theropods, which are planktonic mollusks. And the theropods are divided in two categories: the ones that have shell and are called ecosystem, and the ones that are without shell, which are called chemiopods. But we focus on our research in the first group of specimens because their vulnerability and their key ecological role in the galactic ecosystem. The ecosystem theropods are vital in working because they transfer the energy from the phytoplankton because they feed on phytoplankton, and they are prey of several commercial species and whales and insects. This group is also important in the biochemical cycle because their shells are made of aragonita, which is a soluble form of the calcium carbonate. And when the animal dies, this calcium carbonate sinks to the deep ocean. So this process is essential for the long-term carbon sequestration and for the regulation of the global pH there. And another important reason for this group of species is that they are continuous of change because the shell is made of aragonita. So if we detect changes in the calcification, it's talking about the cells of the pelagic process. Next one? Okay. So if we want to understand the importance of our research, we must look to the Atlantic Ocean. We know that the ocean acidification is a global phenomenon, but we also know that the ocean that is of the ocean acidification are not the same in all the systems or all the ecosystems. So in this regard, the South Atlantic is one of the main players because this region acts as a national CO2 update. This region also functions as a transitional geochemical zone because we can detect the emission of CO2 in latitude higher than 35 degrees and south of 40 degrees in latitude, the system is updated zero. So this all the ecosystem that are in the South Atlantic support highly productive ecosystems. But the important of this system is not only ecological. The systems are also important for the economy, the regional economy, because they sustain a valuable fishing industry. And the main fishing of the fishing industry depends on the zooplankton prey. Depends on the zooplankton prey. And these stimulates are not that because the theropods are one of the key prey of the industry, but we have limited understanding of the effect of social acidification on the theropods. So we don't know how this economically important of this concern will respond to the phenomenon of the ocean acidification. Okay. Well, so we start the knowledge. We select two working ecosystems work in the space. We work with in the Active University of Communication, which is located in the University and we also select the research as which is located between 54 and 55 levels. So I research teleport from these two ecosystems, and we perform our activities in studying these ecosystems. So in the following section, I will detail all the activities that we developed during my stay there, the preliminary results that we got, And the more important things, the skills that we develop through the college. Next one. So the activities that we develop are four main activities. We started with the customer identification of the robot. For this, we use microscope. Then we developed the method for assessing of the chain activity. We prepared the shell for scanning electron microscope, and the last activity was to create a technological scheme of shell dissolution based on the recognition of significant hypotheses. For this, we use pouch information. Well, when we were working at the microscope, we realized that we have two different types of teleport. You can see in the image, that we have a larger group of specimens, and in the bottom one, you can see that the specimens are fine. So we realized that we cannot apply the same protocol for all the specimens. So we developed two protocols for the larger ones, and we developed one protocol for the tiny directly. And the main difference between the two protocols for the larger specimen is the time that we expose the specimen for the investigational phase. Then we prepare the shell for the scanning electron microscope. Well, this is a very meticulous part because of the fragility of the shell. So we prepare the metal stuff, we coat them with a conductive double sulfate, and then we mount the cathodes with their epical facing upward with fine brushes, with only one to there, and we did this under the microscope, and we finally prepare in the first step. We finally prepare one stuff for the different protocols that we are testing. Well, we achieved a result from all the from the taxonomic identification, we identified three different species of Costeras. The first one is the the other one is the Plata, and the third one is the Plata. Regarding the developing method for assessing the shell dissolution, the protocol B give us the optimal device in observing the shell, and the protocol C was also successfully to take images from the smaller. But it's important to say that the result that we got depends heavily on the concentration declaration and expectability. So this is a very important data metric because we need to be certain about this. Because if you want to be sure that the depollution that we are seeing occur in the ocean and not after the preservation of the content, we need to use buffer ethanol, and we need to be very careful about it. Excellent. Well, the same, we got several images from the same analysis, and some images reveal that we have intact shells, but mostly of them had happily early warning signals of type 1 that were scattering over randomly and over the shell. Some experiments that they are type 1 of information, but they are going into type 2, and we detect experiments in type 2 of information. And this is also an important result. We start a significant number of images because we only detect 2 species, and this was because of the sample percentage. Well, this is an example. Intact shell looks like this. you can see, well, the organic and the inorganic layer was removed from the applied phosphate. So you can see the organic layer only, and you can see that it's a plain surface. On the contrary, when you have a type of dissolution, this thickening layer starts to make a little foam because the calcium carbonate crystals are removed. So you can see that in the shell, you can see a little foam that when the type of solution goes into 1 to 2, the foams are becoming deeper. So this is the signal that the solution is continuing and exploring deeper on the structure damage. Well, this is maybe one of the most important slides. So here I tell you that from all the activities that we develop, I gain skills. So from the taxonomic identification, I learned how to identify taxonomically the species. I learned also how to prepare specific protocols for the shape. How to modify it considering the different types or the different sites of the document. I also learned how to apply and implement the trend analysis. I also learned how to recognize the shape dissolution with the images and how to link this dissolution to the aromatic solution state. Well, in my state, Nina was developing another project, so she kindly invited me to collaborate with this project, and I accept. So we developed other activities that they are not in the main plan. So it was important because I also gained skills from this new activity. She was doing ocean salinity assessment experiment. Yep, same time. No, we
Sorry, Georgina, do you want to go back a couple?
Yeah, please. Yeah. Okay. So Nina was developing ocean and community experiment with different disciplines. So I collaborated with this project with her. So I learned how to set up the different experiments. But for developing this experiment, we need to collect the animals. We work with the National crab, with the juvenile, and we also work with the megalophiles crab. So we collect the animals, we learn how to the feeding protocols of this animal, and we also develop activities related to the system management and how to setting up different treatments of ocean animals. So I learned how to design the ocean experiment. I learned how to handle with life organisms. And I also learned how to develop control technical systems. We also work with Copperpot and with Reef. Well, maybe one of the most important things that I learned for this day is that all the protocols are all the
Is that good? Or do you want to go up forward one more?
Yeah, because we are having problems with internet.
Oh, okay.
Okay, so looking ahead, maybe this day was very important with me, for me because now in my home institution, I can continue doing purpose based in Argentina, and I can continue evaluating sensitivity to the ocean spaces. Now I can develop ocean economic experiments, and this is very important because to our knowledge, nobody else is doing it now in our country. So this is very important because now we can establish research lines focusing on this kind of activity. This is maybe also important because there are some enthusiastic doctoral and postdoctoral students that maybe can take advantage of this. And it's also important to say that our results are not isolated. Our results are in full alignment with the Peer2Peer program. So this allows us to take a powerful synergy of expertise. And as an overall assessment, we were thinking about degrees with Marinas, and we agree that this tool was a mutual benefit because we can successfully exchange the knowledge and idea, and our results or our findings are integrated into a real collaborative framework. And from this experience, acts as a expanding of my research, and it's also promising for long-term competition quality. So I would like to thank you for their attention. That's all.
Wonderful. Thank you so much for your presentation, Georgina. That was so great to hear more about what you've been doing. And I always love to see the SEM images. They're so cool. So great to see the amazing diversity of life that you were able to capture too.
Yeah.
Yeah, incredible. So at this time, we'll open up the floor for questions. So feel free to put them in the questions box if you have any for us. But I think I'll kick us off with a question to start. So this is kind of a general question, but I know that there are many current peer-to-peer mentees and mentors in the room right now. Do you have any advice for people who are just starting to participate in this program and maybe hoping to apply for this scholarship?
Well, I think that this is a very important opportunity. It was a very important opportunity for me because I really fund my research curriculum. I am not, I am a middle career researcher, so now I can establish a new research line in my home or in my country because nobody else is kind of here. So I think that one of the most important things from this experience was because now I can apply all the knowledge that I gained. I continue in Dutch in Mina, so we can continue collaborating and working together. In fact, we have plans to work together in the future and continue with this. So I think that it was the most important thing. Not the data or the results, but the experience itself it was very important.
Yeah, I definitely I think that the experience definitely can be more valuable than the research sometimes because it leads to so many other partnerships and learning experiences. So I love that answer. Thanks. So it looks like we don't have any other questions in the chat right now. But again, I would encourage you all to, if you have questions that pop up later, reach out to me, reach out to Georgina, and happy to keep the conversation going. Oh, wait, one question maybe. Let's see. All right. Yes, we do have a question from Alejandro. Hello, I liked your presentation. I'd like to know if it's a requirement that the samples be observed under a scanning electron microscope to determine the type of the solution and the average pH level in the area. I'll copy it. You see it.
I think that is the only way and the average pH in the area, I don't remember, but we are now analyzing the result. We are trying to align the equation where we got the solution with the pH and the omega alanine result. We are doing it in collaboration with Pier2Peer. Pier2Peer. So it is our next step to first to finish to analyze the images and then to match the data, our biological data with the biochemical data. But the pH level, I don't know.
Excellent. Thank you, Georgina. And it looks like we have another question from Alejandro. At what depth did they obtain the aragonite saturation horizon?
We obtained from the first 100 meters of the volume. But from the samples that we analyzed from South Georgia, we have certified samples. So the origins were in an particular depth, not the whole volume.
Excellent. Thank you.
That for me is very important because for the Argentinian ocean competition, it's the first time that we got this kind of report because Ina was previously working in this contractor, so we have previous evidence that we find the ocean solution today. But for the the ones related to the access, it is the third report for solution operation. I forgot to say it, and I think that is important.
Awesome. Great. So let's see if we have more. Okay. No more questions in the chat, but Alejandro says thank you. And I think that's bringing us to the end of our conversation today. So thank you all, everyone, for your presentations, your thoughtful questions and contributions. We're coming to the end of our program today. But before we close, I'd like to extend a huge thank you to our two speakers, Dr. Georgina Cepeda and Dr. M. Anand, for sharing these amazing examples of exciting research and also international collaboration that's happening through the peer-to-peer program. We had two excellent presentations today, and I'm excited to hear more from these two researchers as they continue in their fields. And I'd also like to thank our audience for joining us today to celebrate the achievements of our peer-to-peer scholarship recipients. Thank you all so much for joining, and I will see you next time.